Good morning, everyone. Welcome to the September 2025 Zoom Citrus Seminar. Thank you for joining us. First, I would like to thank Deborah for her help in our Hendry County Extension Office in LaBelle. Today's program offers one CEU for pesticide license renewal and one CEU for certified crop advisors. If you need CEUs, email me your name, email address, and license number. The title of the seminar presentation is Harnessing Host Tolerance for Long-Term Management of HLB in Citrus. Our guest speaker is Dr. Nabil Killiny, Associate Professor in Plant Pathology at the Citrus Research and Education Center in Lake Alfred. Dr. Killiny will uncover physiological, molecular, and genetic elements in tolerant citrus varieties that limit infection or disease progression caused by HLB. Dr. Killiny will also identify threats in tolerant citrus varieties that deter or limit infestation by the Asian citrus psyllid. And he will explore conventional breeding and other breeding approaches like gene editing to introduce and stabilize tolerance traits in commercially important citrus varieties. Dr. Killiny, it's all yours. Please go ahead. Thank you.
Thank you so much. Thank you so much, Manji, for the invitation. I'm always happy to participate in this program, and I hope my presentation will be likable by everybody. And I will actually talk mainly about my program, however, based on a lot of work done by other researchers. So just to start, well, citrus greening disease or Huanglongbing started in 2025, and we start to have a decrease in the citrus production, as you can see in the first figure. And this decrease continue decreasing until now, with really a very bad situation, about maybe 5% of what we used to have in 2004. The end is dead tree, like you can see here also in the last figure. But it takes time until we have the tree completely dead with that way. Well, just remind you with the symptoms, I'm quite sure everybody knows, but that's the key for me to talk about the tolerance or potential tolerance to the pathogen and to the insect vector. Chlorosis in the leaves, so it's less photosynthetic activity, and yellow vein. We also have cork vein, or it's actually kind of death, cell death inside on the leaves. The fruits is unequal, like you can see, and the greenish from outside. And here is the main problem, is the fruit drop, premature or pre-harvest fruit drop, which actually is the main damage here we have. Why we have all of that? Because the root systems get decreased so much. You can see the root system here in the infected compared to healthy. These two trees have the same age and treated the same nutritional treatment and everything, except this one is infected with the pathogen. So less root system, so less nutrition goes to the tree, and we start to have all these symptoms. The major problem is this disease is not caused with normal pathogen. It caused with the most destructive pathogen, which is vector-borne pathogen. So if we have a bacteria or a virus infect the tree, and this virus or bacteria is not insect transmitted, the problem is much, much less than we have now. And we will discuss that in the upcoming slides. It's very simple. No vector, no disease. So imagine that we don't have Diaphoreus cetri in Florida. We will never have this situation of Huanglongbing. So what is different in the patho system in citrus greening? So citrus greening patho system have three main players. Have the pathogen, which is bacteria, is not available yet in culture, and this actually limit a lot of studies and limit even confirming that this is the right pathogen of HLB. And this is why it's candidatus. So the genus candidatus in bacteria, in bacterial classification, meaning that it is not confirmed yet as a pathogen to the disease. In order to confirm, we need to have the pure culture of the bacteria, infect the tree, produce the symptoms, isolate the pathogen again, cultivate, inoculate in another plant, and we produce the same symptoms. We cannot do that. Bacteria is not available in culture. And all what we have is just a trial, and the bacteria is very small portion among microbial community. And this is why it's probably the microbial community, including the Candidatus liberibacter asiaticus, are the pathogen, the real pathogen. Anyway, the host is citrus, so the bacteria is very specific. So the bacteria just infect citrus and citrus relatives. And what is really worse is this bacteria is transmitted by insect vector, the Asian citrus psyllid, Diaphoreus cetri. Diaphoreus cetri is hemiptera or hemipteran insect, which means that the insect feed on the phloem, on the phloem sap, because the insect sucks the sap from the phloem. So it has a stylet, piercing stylet, that can reach the phloem, feed, sucks the sap, and the bacteria also. And from here, we start to have acquisition of the pathogen, and when the insect fly to another healthy plant, start to feed, and the first step of feeding of hemiptera is to inject saliva. And while injecting the saliva, the insect injects the bacteria. And like that, we have the spread of the disease. Again, no vector, no disease. If we don't have the vector, we don't have the disease. Why? Because the bacteria is phloem restricted. It is not on a foliar bacteria, for example. It's not on the surface. No, it is in very specific tissue, and it needs a specific vector that can reach this tissue, which is the phloem, and inoculate the bacteria. Just to recognize the vector more, as you can see here, here is laying eggs. So here is the adult, and the adult lay eggs in the new shoots. So new shoots are very important. And there are a coincidence here. The insect loves the new shoots, the small leaves, but also the bacteria migrate all the time to the new shoots. So the insect lay eggs. After hatching, the eggs give nymphs, and just for your information, nymphs feed continuously. It feeds much, much, much more than the adult. And we believe that here is the step of the acquisition. So the insect acquires the bacteria while it's nymph. And after emerging, like the third photo here, the adult can fly to another tree and inoculate. So most likely the acquisition happen while the insect are nymphs, and the inoculation while the insect are adult. And as you can see, we have 5 different instars of the nymph. And here is how the egg look like. It's very important also to know that when we have a bacterial pathogen infect a host, but this bacteria is insect or vector transmitted, insect or arthropod, or that means the bacteria have dual lifestyle. For example, our bacteria live in plant host inside the phloem sap, and also live and propagate and grow inside the hemolymph of insect. And this is actually really two completely different lifestyle. And this is why the bacteria has completely different two major transcriptome in order to live in the hemolymph, like you see here in the up chromatogram, or the phloem sap, like in the bottom chromatogram. In the phloem sap, you can see the major peak here are sucrose, and this is normal. In the phloem, we have the sucrose created from the photosynthesis. And inside the hemolymph, the major disaccharide is not sucrose, it's trehalose, which also makes sense for insects. So the bacteria can manage and can feed in both of them. There are some common compounds, which is actually very important for the bacterial growth. But in fact, the bacteria also can use alternative compounds. Compounds exist in phloem sap, different than what it exists in the hemolymph. So this is how the pathogen is very complicated. It's not simple bacteria. It is bacteria with dual lifestyle, although the genome is very small. But actually, because the genome is very small, bacteria doesn't have a lot of enzymes to create things. So the bacteria depend on the nutritional surrounding media inside the hemolymph and inside the phloem sap. So the bacteria can acquire a lot of compounds and deal with a lot of compounds as nutritional, as carbon source, and then affect the plant by the metabolites, and we have the symptoms, or the plant response. So we have the symptoms. So just to start, two major points we need to keep in mind, and it's very important. Many of the most destructive diseases in citrus are vector-borne. And I will just remind you with citrus tristeza transmitted by brown citrus aphid, citrus stubborn caused by Spiroplasma citri and transmitted by the leafhopper circulator hematoceps, CVC or citrus variegated chlorosis caused by Xylella fastidiosa and transmitted by the sharpshooters, the xylem feeder sharpshooters, because this bacteria is specific to xylem. So when we have the word vector-borne combined with the bacterial pathogen, we have a serious problem. And actually it make history. So there are a history of the CTV and CVC. And now we are living this history with citrus greening, citrus or Huanglongbing. That's one point we need to keep it in mind, because it's very important when we want to find tolerance elements and try to come up with tolerant varieties. Second, because the tolerant varieties could be tolerant to the vector. If the variety tolerant to the vector cannot feed and will not transmit the pathogen. So it's very important to know that. And second, as I mentioned, the bacterial pathogens that are transmitted by insect have a dual lifestyle. and it has effect in the plant host and insect vector. But for sure, plant is more important for us because it's citrus and it affects the production. In order to have citrus greening disease, in order to have the disease created, and we start to have the symptoms, and the trees start to suffer, does a lot of responses. Sometimes the auto response of the tree kills the tree. And this is the case actually we have in citrus greening. In order to have that, we need to have 3 different interactions or tritrophic interactions. It should be happen in order to have the disease. So the pathogen host interaction, the vector pathogen interaction, and the vector host interactions. So all these interactions are very specific. And when all the interaction happen, we have the disease. For the bacteria have a challenge here because the bacteria need to change its transcriptome, while the transition from host to vector and from vector to host. 3 interactions, it's tritrophic interactions. Okay, so there are factors affecting this interaction, correct? So the vector-borne disease complexity in general, one more partner in the pathosystem is the vector. So it is not only 2 partners, which is the pathogen and the host. No, there are a vector here. The pathogen need to interact with the host and the vector, both of them. The pathogen should be able to grow, in our case, to grow in both insect and plant. And the successful interactions are required to have transmission and the spread of the disease. What are the factors affecting all these interactions? If we look at the pathogen with the host specificity, and our pathogen seem to be like very specific to citrus and citrus-related species. Host susceptibility, and unluckily, all citrus varieties are susceptible. Some of them are tolerant for certain extent, but it's not to say that we really have a tolerant variety in hand. But we still categorize few varieties like a little bit tolerant. Maybe we need to say a little bit. And the pathogen virulence. And here we don't know so much, by the way. How many strain of the bacteria is there? Does the bacteria has some mild strains, avirulent strains? And it is very important to know all this structure because maybe we can do something if we understand how many strains we have in Florida, for example. How many strain of them are very severe and some if there are any mild. So we need to compare, for example, and see the mild strain, what is missing. So we'll understand more about the pathogenicity. Then the interaction of the host with the vector. Host preference. Yes, also the insect have preference, and the insect just follow the bacteria, and you will see that. Just follow the bacteria. So this insect, almost monophagous, which just feed in citrus and citrus relatives. Feeding site is very important. Insect feed on the leaves, mainly on the leaves. Tissue preference is also important. The bacteria is restricted to the phloem, so the vector is feed on the phloem in order to pick the bacteria and do the transmission. These are the factors. And finally, the interaction between the vector and the pathogen. And here actually there are very specific interaction in order to have the pathogen transmitted by the insect. So the bacteria interact specifically with the gut of the insect, do attach to the gut and even form biofilm. We will not talk about this interaction today. Today, I focus mainly in the host preference on the interaction between the host and vector. And also we'll talk about the pathogen and host. Just to make the long story short and the huge amount of research in the interaction of the insect, Asian citrus psyllid with citrus, we have this information, very specific information. Feeding sites, preference, and oviposition. That's very important. Very important, because if we can interfere with the oviposition, for example, so we'll not have the eggs attached to the new shoots, will not have hatching, will not have nymphs, will not have adult, we will not have transmission. So Diaphoreus tribe prefers the young, tender citrus flush for feeding. That's make a lot of sense because it's easy to feed, easy to inject the stylet, and it is very nutritional also. But this is not the main reason. The second reason is the female can lay the eggs on the new shoots easily. So it is also for oviposition. And this tissue is high nutrient content. So the insect adult, the female, will secure its babies that there are a lot of good food there. So new shoots is better. So they put there the new shoots, the eggs. But again, the movement of the bacteria move with the new shoots and just go to the new shoots. Actually also move to the roots while there are new shoots in the roots, new growth in the roots. So certainly there are bacteria, and then the hatched nymph will feed and acquire the bacteria. That's number one. And number two, psyllid is not different than other Hemiptera, the order of insect that include Diaphorina citri. Hemiptera have a photoreceptors tuned to the UV, blue, green light, and yellow. And this is very similar actually to the young leaves. Okay, so this color attracts psyllid, especially the light green and yellowish, like you see in this paper. The new shoots kind of yellow or very light green. And the insects, the Hemiptera, doesn't like the purple color because it doesn't fail, it actually fail outside their peak sensitivity range. And this actually make anything having this color is not attractive to psyllid at all. So psyllid doesn't like the purple color. Actually, I don't like this color too. Anyway, so now we have 2 points here. Yellow or light green can attract, purple can deter the psyllid away. And this is what we can see here. So started by Lucas Teleneski group, and they make this nice device. It is yellow, so it's really attract the psyllid. And inside this attraction, this device, let's say poison, whatever this is for the insect. So this is actually the strategy which called attract and kill. But we need to use these devices. We need to change these devices, and it's maybe costly. What if we make it sustained? What if we create some citrus to be attracted by psyllid, and then you use those as a border to the groove? Here we used a system, it's called CTV vector, to knock down phyotine desaturase. It's the precursor of carotenoid. And when we do that, when we silence this gene, we have this phenotype, like you see. So this phenotype called photo bleaching. So you can see still have green and dark green, but it has a lot of yellow there. This plant is very attractive to psyllid. And you can see here, the black columns here are the BDS plants. And you can, here is all the in light and in dark preference assays. When we release psyllid in cages, have plants from BDS and normal green plants, and then count the landing, how many insects landed onto the BDS plants and how many landed in the green plants. You can see here, here is the average. The majority goes to the BDS, to the photo-bleaching phenotype. In dark, more or less, there are no really significant difference, which mean that the visual cue are very important, even more important than the volatile that attract psyllid. When we look at the volatile, released volatile, so it is really enhanced. So there are a lot of differences. And when we look at the nutrition and the phloem sap, so here is the phloem sap nutrition, there are actually a lot of differences there. But the plant grow very well, and attract psyllid. So we can say that these plants, when we silence phyotene desaturase, we have a lot of volatile released from the leaves. This volatile, it's actually help for the olfactory cue. When we look at the pigments, so pigments are less, right? Because we knock down or we silence the carotenoid pathway. And this is very important to say. Silence meaning reduce, because if we try to knock out completely, the plant will die. The plant cannot live without pigments. So photobleached phenotype, that's a visual cue. That's also attract psyllid. And I showed that metabolite also is very good. And this actually, it's very good also for the food for cells. So psyllid loves these plants. So we can use it as a potential trap crop. But actually, this is not enough. We can also have the same plant, photobleached plant, produce small RNA to make RNA to psyllid. So when psyllid loves this food and start to feed, it will also get some poison. So small RNA in this case is two genes, one gene called AWD or abnormal wing disc, and the second is wingless protein. These two genes are implicated in the wing formation. And you can see here the malformation of insects feeding these plants. So we can actually stop the insects from flying away. And this also is very important to stop the transmission. This is very sustained because the plant will continuously produce a small RNA. And the plant will continuously having this phenotype. So attract and kill, even in different generation. If we keep one population in trees and keep counting, we will see that generation after generation, the population getting very low. In just 2 or 3 generations, the plants are clean. But also, there are effects in laying eggs. That's very normal because the adults are affected severely. So will not have normal mating and normal laying eggs. So now we change it from attracting only from trap crop to also killing the vector. We can produce wing malformation, so limited flight, high mortality, short lifespan, and low fecundity, like I showed, much less number of eggs. This work has been published in horticulture research. And if you are interested in having much more details on it. Second sentence I mentioned that psyllid does not like this color, purple. What is the evidence? The evidence that in Christopher Vincent group, they start to spray red or kind of similar color of clay, kaolin film, to make a kaolin film. And as you can see, he showed that there are no, it does not interfere with the photosynthesis. And this is very nice, very good. We also showed that later in transgenic blend. But the point, and this actually also tether psyllid away, repellents to the psyllid. That's very good. But we're in Florida, we have a lot of rain, and the rain can remove the film. So why we don't just make it sustain, make it, and this actually with transgenic. Here we just overexpressed transcriptional factors that enhance the pathway to express anthocyanin, and the anthocyanin gives this color. We did photosynthesis study, and the photosynthesis actually is not changing dramatically. So the changes are very low and doesn't affect the efficiency. Now, let's see again, like I showed with the photobleached blends, you can see here the expressed anthocyanin expressed tree, preference, released 100 insect. So the majority of insects just go to the green normal tree. So it's really working to tether the psyllid away. But maybe also volatile. We looked at the volatile, and yes, we found some different here. Here is the average. But when we do the BCA, there are no big differences actually. There are no separation, let's say. So we don't know exactly. To answer this question, we made the preference in dark, like you see here. Here is the cage, 2 plants inside, released 100 insect, and there are no significant. Sometimes the majority on the purple trees, sometimes the majority on the green tree. So this actually also shows that the volatile have a minor role, and it's mainly color attraction or color cue. Same thing here, if we use paper in different color, like light green, dark green, purple, dark purple, and then release the insect, the majority of insect actually land on the paper with the light green and dark green. So it is really visual. It's not only that, when we enforce insect to feed on those plants by cage insects on purple plants or normal plants, and then you can see it's affected. The lifespan is shorter and the survival are affected severely. And when we collect the phloem sap from normal plant and infected plant, so the phloem sap contain the anthocyanin, actually and let the insect feed through artificial diet, higher mortality with the phloem sap that contains anthocyanin. When we use anthocyanin like authentic standard, yes, we have also high mortality. So purple leaves tether ACB because of the visual cue, not because of olfactory. And also anthocyanin is toxic to ACB. So can use those plants to tether the psyllids away from citrus. Well, now back here, again, we have tritrophic interaction. We talked about how we can probably find some tolerance from the interaction between citrus trees and vector. And let's now talk about host pathogen. Again, here is the interactions. And now we are here, the interaction of pathogen with the host. And here we have the host specificity, host susceptibility. Host specificity, so we know that the bacteria is very specific to citrus and citrus related. Host susceptibility, all citrus are susceptible, with few varieties show some tolerance. A little bit, like I said, I will add a little bit of tolerance. So let's see what we can do here. I will, instead of just talking about the cellular responses to the pathogen, I will actually just compare between very susceptible one, which is Hamlin, sweet orange, Hamlin, and Barson Brown. Why Barson Brown? Barson Brown looks like shows some tolerance. And actually growers asked us to look at why this tree looks much better. And why this tree live much more. For example, 15 years ago, it was trees of Hamlin, and those trees were next to each other. And still, the Barson Brown trees look good and still alive and show tolerance. So comparison here, it's from the nature, from trees, not single tree, trees from 5 different locations in Polk County. It looks like that. It looks very, very good tree. Let's understand why this tree survive, and we can call it survivor. So why we can this tree survive HLB? We compared here between Hamlin and Barson Brown, but we also got another variety called Rubel, and this Rubel, the 3 of them are early fruiting or early flowering. Okay, so they are the same category of sweet orange. And when we do the preference, we notice it's not statistically significant, but it's a little bit less of insects goes to the Barson Brown. And when we do the infection, the CT, it's higher in Barson Brown, also is not statistically significant, but we can see the same trend. Here is after 6 months, and here is after 12 months. We have the same trend. More or bigger CT value, less bacterial population. Okay, so it seems that bacterial population a little bit less, but regardless, it doesn't really matter because we know that bacterial titer in a certain point doesn't make any sense. Low bacterial titer equal high because the bacteria trigger the responses, the continuous responses of the tree, and this response is actually what kills the tree. This transcriptome, it's a very, very nice study, but all what I want you to see here is a volcano for comparing the transcriptome of Hamlin, Rubel, Barson Brown after infection with the bacteria. Let's see here, for example, the down-regulated genes after infection in Hamlin, it's like 280. Here it's 470, but in Barson Brown, only 60. If we look at the up-regulated genes, much higher in Barson Brown, like double of what we have in Hamlin or Rubel. So there are a lot of pathways actually are enhanced, and there are a lot of genes changed here. Let's see also here when we compare infected to infected, Barson Brown to Hamlin. So Barson Brown have 450 genes upregulated when we compare Barson Brown to Hamlin. And here is with Rubel. And here is Rubel with Hamlin, it's only 200. While again, Barson Brown with Rubel, 300 something. So there are distinguished. That means the response of Barson Brown are different than other sweet orange varieties. And let's look at the different genes. And when we look at the most affected genes, beautiful here, you can see the Barson Brown, it's really higher in a lot of genes. That's all salicylate. So salicylic acid actually is much more. Okay, expected. Flavonoid, okay, expected. Caffeic acid, which is phenolic compounds, okay, expected. And this is what we know, actually. That's the response of the infected, the response of citrus when it get infected with the bacteria. It's just higher in Barson Brown. You can see here also, it's always higher, higher, higher when we compare. But when we compare everything here, healthy and infected, you can see the bigger one is Barson Brown in the total phenolic content and total flavonoid content also. Okay, so it's just higher in those. Okay, higher in total phenolic content and higher in total flavonoid content. Let's see something else. Let's start with the transcriptome. Transcriptome also shows that the response here is not similar to each other. So let's see in biochemistry. When we look here in A, here is Hamlin, Rubel, and Barson Brown. And here is the Ross, which is actually here is the superoxide anion. So you can see that there are response to the infection. Response. But overall, the lowest are in Barson Brown. When we look at the hydrogen peroxide, more or less the same. The lowest one here in infected Barson Brown compared to the infected Hamlin and Rubel. So it seems that the Ross reaction is moderated. So it's a little bit less. That also makes sense because hyperactivity of the Ross reaction actually kills the tree. Here are the same, but with staining inside the tissue, and it is a very similar results. Much less here in Barson Brown. Look, it's almost similar to the healthy. But here with the OT, it's actually is not very similar to the H2O2. Here is another way, is fluorescence. And this also for the O2, and you can see here in the infected, this is Hamlin. In the infected, you can see the fluorescence goes inside the merge of the leaves, which shows that superoxide anion is actually much higher. Compare that with Rubel. Oh, Rubel is much less. Compare that with Barson Brown. It's really almost clean. So that actually means that the Ross reaction is less. Ross is now let's look at the volatile. When we look at the volatile, real volatile, released volatile from the leaves, we can see this kind of pattern which have different clusters. And this is very important. These genes, these actually volatile are very important for attraction. And it is not because it's high that means the plant will be attractive, because it's a mix. It's usually mix of volatile, blend of volatile, which can attract psyllid or deter psyllid. And when we look at the genes related to the pathways in the transcriptome, you can see here infected Barson Brown is very high. So it is actually do something very important. Something very important. So we believe that although we didn't find significant difference, but we found that lower insect population get attracted to Barson Brown. So it's maybe less insects and less transmission. Ross is reactive oxygen species. Then let's also look at something else. And it's very classical, very classical. HLB callus accumulation or callus deposition. Starch content, higher starch content in the leaves. In the normal case, the starch actually stored in the roots. When we have infected with HLB, it is a contrast. We have the starch in the leaves and actually also goes to the fruit and cause. One of the reasons that we have fruit drop is the accumulation of starch. So let's look at the leaves first. Here's the healthy. So the healthy, there are no starch stored. Infected Hamlin, the very susceptible, it's very black or blue, like you can see. Rubil also more or less similar, but Barson Brown much less. So less starch. When we look at the callus deposition or callus accumulation, you can see that callus increased after infection in everything, but there are no significant difference after the infection in Barson Brown. We look here in the tissue. Here is actually the tissue stained with aniline blue. This is why the photos are blue, by the way. So you can see here the accumulation or the callus deposition in the phloem. This is the bark from inside when we peel the bark and we stain. And here is a section. You can see here also inside a section of a stem. More or less rubil and much less in the Barson Brown. That's very nice and just agree with all what we know and what we expect. We expect that if we have tolerant variety, it will have less callus and less starch. When we look at the metabolites, the main difference we can see is the disaccharides. So the disaccharides is usually sucrose in our case because it's phloem same. So sucrose even getting higher after the infection. And this maybe have something to do with the starch. This why we have less starch, because the disaccharides, the sugar, sucrose didn't get into the starch. So the take-home message here is citrus responses to HLB are not very specific, and that's something I really want to emphasize. All what we talked about, it is responses to biotic and abiotic stresses. Everything, from ROS, from callus deposition, from total phenolic compound, from total flavonoid, everything we talked about, it's actually more or less related to common response to the biotic and abiotic stress. And second, the tolerance of Barson Brown that we see, it's driven by many things in the defense response. So it's a cascade of pathways and genes. And this is very important because this fact emphasizes that breeding is very important, because with breeding, we can have this trait. If we keep having hybridization, maybe we can have varieties that similar to Barson Brown, but a little bit even higher, so it would be tolerant to the HLB. In contrast, transgenic or even CRISPR, it's actually depend on one or two genes. And what I can tell that this disease is not like gene to gene. It is not like one single gene. If we target, the plant start to be tolerant or start to be resistant. And it is obvious from this work and many other work, like it's actually many genes. We cannot get all these genes all together. And if we keep working in one single gene, let's say enhance or knock down ROS, this is exactly what we will have. Imagine that one gear in the watch, you remove it and you put instead a much bigger one. The watch will not work actually, and the plant will die, because that's actually the normal response to all stresses. So in order to have tolerant to HLB, we don't need to have a plant susceptible to abiotic stress. So It is very difficult, in my opinion. That's my own opinion. And what we need from this work and all other published work, well, we need to have less Ross reaction, less callus deposition, accumulated starch should be also less, more flavonoid and more phenolic compounds. How come we can do all of that with transgenic? Negative regulator is not very well characterized, and also it is part of a lot of big machine, like NBR3 and NBR4. And working on it, it's just the salicylic acid compound will be affected. What about the other? So in my opinion, traditional breeding, it's a key to have a true tolerant variety or resistant variety to be working in the field. So the conclusion of this work and this presentation is we can use the biotechnology to offer sustainable solution based on something like the attract and kill and repellency. So vector is very important. You guys, we don't need to forget about the interaction of the vector with the plant. All the focus on the bacteria with plant. Also, if we can just get rid of the vector, we will not have the disease, I promise. And the effective control of insect vector, like with this method, maybe it's very critical step toward solving the problem until the breeder provide us with tolerant varieties. And this is my own opinion. And by that, I finish my presentation. I need to thank my group. It's a lot of people here worked over the years in many things. My collaborator, Manjul Dot, Sid Gauda, Lucas Teleneski, and Christopher Vincent. And I thank you, and if you have any questions.
Thank you, Dr. Killiny. Any question or comment? Everybody will be planting Person Brown.
No, Person Brown, it's also, I think it's very good to use for hybridization to create a good sweet orange, especially for juice, as a source for hybridization for breeding. But Person Brown have some problems. For example, the oil content in the juice, it's more than 0.035% that allowed by USDA. So that's actually, it's kind of problem here. So the juice, it has some issue. But Person Brown, in my opinion, if we just focus on it with more breeding or hybridization with some other tolerant varieties or finger lime, for example, maybe, or something like that, we probably can have some traits and we can have tolerant varieties with fruits and juice meet the commercial and the USDA criteria.
Okay, again, if you need CEUs, send me an email with your name, email address, and license number. So thank you very much. And
Thank
we
you.
will see you next time.
Thank you so much.
Thank you, David.